Hi, hello everybody. Yeah, thank you very much for coming to my talk today. Also, I would like to thank Immuno-Oncology for this event and for this invitation. So my name is Jia Shen. Currently, I'm working as an assistant professor in Department of Medical and Molecular Genetics, also medical science program in Indiana University School of Medicine, Bloomington. So today I'm going to talk about how we target the tumor intrinsic pathways trying to enhance the cancer immunotherapy. All right, first of all, our research interest. As we know, cancer is a major threat to human health. In order to combat cancer, we human beings have a long history for that, including surgery, radiotherapy, chemotherapy, and targeted therapy. In 2010s, immunotherapy emerged, and since then, it has revolutionized the whole cancer treatment field. So the concept of immunotherapy is to use our own immune system against cancer. However, there are still a lot of patients, they have immunotherapy resistance for different reasons, which I will talk about it later. To this end, we are very interested in targeting the cancer cell intrinsic pathways, trying to enhance the immunotherapy efficacy for the cancer patient. We know that in the tumor, there are at least two types of cancer cells. One is the regular cancer cells, or we can call them differentiated cancer cells. And the other type of cancer cell is the cancer stem cells, or some people call them tumor-initiating cells. So during my postdoc research period in San Diego, California, my research focused on targeting the regular cancer cells. And currently in IU Bloomington, we are focusing on targeting the cancer stem cell signaling pathways to enhance the cancer immunotherapy. So today, in the following time, I will talk about both of those two directions. All right, so there will be two parts in my talk today. First, I will talk about how we target the regular cancer cells for cancer immunotherapy. Then I will switch the gear to talk about our research on targeting the cancer stem cell signaling pathways. All right, so FBXO44 pathway in regular cancer cells and its targeting can enhance the cancer immunotherapy. First, the background. Immune checkpoint blockade (ICB) therapy is a major type of immunotherapy that has been approved by FDA to effectively treat multiple cancer types. So this immune checkpoint blockade therapy can activate the tumor-specific T cells to induce or enhance the anti-tumor immune responses. As you can see here in this figure, on the left, without the ICB therapy, and in the tumor microenvironment, the tumor cell immune checkpoint protein called PD-L1 could directly bind to T cell immune checkpoint protein PD-1, which could cause inactivation of the T cell anti-tumor function, finally leading to tumor escape. In contrast, with the ICB therapy, by using anti-PD-L1 or anti-PD-1 antibodies, so the tumor-specific T cells could be reactivated, finally leading to elimination of the tumor cells. However, there are still a lot of patients, they don't initially respond to this immune checkpoint blockade therapy. We call them primary resistance. Or some patients, they eventually develop acquired resistance to this ICB therapy. So sadly, those patients cannot be benefit from the high efficacy of ICB therapy. So you may ask, what mechanisms cause this immune checkpoint blockade ICB therapy resistance? Currently, we know that low tumor immunogenicity and also the immune suppressive tumor microenvironment are two major reasons for that. What is tumor immunogenicity? The tumor immunogenicity means the ability of the tumor to provoke an immune response. We know that loss of MHC1 expression on the tumor cell surface, low antigen processing and presentation pathway, and also upregulation of the PD-L1 expression all could decrease the tumor immunogenicity. On the other hand, low functional cytotoxic T cell, CTL, and NK cell infiltration, and also high number of immune suppressive cells, such as Treg cells, MDSCs, all could contribute to immune suppressive tumor microenvironment, or we can call them immune cold microenvironment. And in most of cases, low tumor immunogenicity could significantly contribute to immune suppressive tumor microenvironment. Therefore, in order to overcome this immune checkpoint blockade ICB therapy resistance, enhancing the tumor immunogenicity is the key. Therefore, our research question is how to enhance the tumor immunogenicity. I guess we can learn something from the virus. So when the virus infects the normal cells, the DNA or RNA released from the virus that can activate the cytosolic nuclear cell sensing pathways, which include C-gas STING pathway to sense the DNA, RIG-I or MDA5 Marvis pathway to sense the RNA. Both of them can activate IF3, IF7 that are two transcription factors to induce the interferon alpha, beta signaling, and downstream signalings. So those downstream signalings can enhance the immunogenicity of the virus-infected cells by increasing the antigen presentation of the virus-related peptides, and also induce the secretion of the cytokines, which could attract T cell and NK cell to clean up the virus-infected cells. So similarly, we sought to induce a larger number of DNA and RNA in the cancer cells to mimic this viral infection process. So this viral mimicry strategy was expected to enhance the immunogenicity of the cancer cells by increasing the antigen presentation pathway and also the secretion of the cytokines that can attract T cell and NK cell to attack the cancer cells. So the next question will be, how can we get those large amount of DNA and RNA at the beginning? I guess the most straightforward way is just to infect the cancer cells with virus. But at that time, we were not sure about the safety. So we turned to the endogenous materials in the cancer cells. So we thought that the repetitive elements in the human genome might be a good candidate for that. The data from the Human Genome Project suggests that the repetitive elements, or we can call them REs for short, that comprise around 50% of the whole human genome. And those repetitive elements could be further divided into some subgroups, including the tandem repeat, for example, the satellite repeat, and also the interspersed repeat, including SINE, LINE, ERV, and the DNA transposons. And those repetitive elements play a crucial role in maintaining the genome integrity and driving human evolution and diversity. Importantly, the repetitive elements are normally transcriptionally repressed by DNA methylation and H3K9 trimethylation. All right. Based on that, in contrast to several other labs targeting the DNA methylation, our final strategy was to target the H3K9 trimethylation to reactivate the repetitive element in the cancer cells. So the transcripts from the repetitive element could form double-stranded DNA and double-stranded RNA, and then to induce the viral mimicry signaling in the cancer cells. And this induced viral mimicry signaling was expected to achieve enhanced tumor immunogenicity and also immune-active tumor microenvironment or immune-hot microenvironment. And those two can work together to overcome the immune checkpoint blockade ICB therapy resistance. So this is a rationale about our study. All right. So this is a preview of the major findings for this study. So in this project, we identified a largely unexplored E3 big ligase, F-box protein called FBXO44 that can form a complex with SUV39H1, which is H3K9 trimethylation methyltransferase. And those two can work together to promote the repetitive element silencing through H3K9 trimethylation in the cancer cells. Furthermore, we demonstrated that targeting this pathway could induce the viral mimicry signaling and to achieve enhanced immunogenicity and also cell death of the cancer cells, finally leading to restrained tumor growth and also enhanced immunotherapy efficacy. All right, now let's move on to the result part of this study. First, in order to identify the regulators of H3K9 trimethylation in the cancer cells, we performed an image-based RNAi screen by staining H3K9 trimethylation as one of the readouts. The result identified FBXO44, which is a largely unexplored F-box protein E3 big ligase, was a top hit. We found that knockdown FBXO44 could dramatically decrease H3K9 trimethylation in the cancer cells, suggesting that FBXO44 is a regulator of H3K9 trimethylation in the cancer cells. Because H3K9 trimethylation is a repressive epigenetic suppressor for the repetitive element. So as expected, we identified an extensive and dramatic upregulation or reactivation of those repetitive elements in the cancer cells, suggesting FBXO44 could regulate the H3K9 trimethylation mediated repetitive element silencing. As I mentioned earlier in the background, reactivation of the repetitive element could activate the viral mimicry signaling in the cells. As indeed, we find that the double-stranded RNA, as here stained in green, and the double-stranded DNA, as here stained in red, were dramatically accumulated in the cytoplasm of the FBXO44 knockdown cells. Also, we found that the nuclear cell sensing pathway, interferon pathway, and chemical pathways were activated upon FBXO44 knockdown, including the upregulation of the nuclear cell sensors such as cGAS, STING, VIG-I, MDA5, and MARVELS, and also the downstream interferon alpha, beta, and the cytokines, which include the CCL5, CXL9, and CXL10. Notably, those three cytokines play a big role in attracting T cell and NK cell into a tumor. Taken together, these data suggest targeting FBXO44 pathway can activate the biomimicry signaling, IFN alpha beta signaling, and also can enhance the immunogenicity of the cancer cells in vitro. Next, we sought to ask what is the molecular mechanisms behind this FBXO44's role in regulating the repetitive element silencing. To this end, we performed a mass spec experiment, and we found that a bunch of proteins related to DNA replication and heterochromatin assembling bind to FBXO44. So, a long story short, by using a series of biochemical and molecular biological experiments, we proposed a working model as shown here. So during DNA replication cycle in the cancer cells, the FBXO44 protein could directly bind to histone H3K9 trimethylation, and then to recruit a complex which include RBPP47, COL4, Nuda complex, and SUV39H1. And this complex can utilize SUV39H1's methyltransferase activity to modify H3K9 trimethylation onto the newly synthesized histones, and then to silence the nearby repetitive element. So when you're targeting FBXO44, the H3K9 trimethylation cannot be deposited correctly, finally leading to de-repression of those repetitive elements. Also, the following experiment demonstrated that targeting SUV39H1, the key component of this complex, can phenocopy FBXO44 targeting. All right, so earlier I showed you that targeting this FBXO44 pathway can activate or enhance the immunogenicity of the cancer cells in vitro. Next, we would like to investigate the in vivo effect. To this end, we inject the breast cancer cell 41 cells into the immune-competent biopsy mice, and we found that knockdown either FBXO44 or its downstream effect SUV39H1 could restrain the tumor growth. Also, we found that the expression of the MHC1 pathway were upregulated in gene knockdown tumors, suggesting the activation of the antigen presentation pathway upon this gene knockdown. Also, the immune cell profiling data showed that there are more functional CD8 T cell and NK cell in the gene knockdown tumors, suggesting that targeting this pathway can enhance the infiltration of the CD8 T cell and the NK cell into a tumor. Actually, that enhanced infiltration, is a good sign for overcoming the immune checkpoint blockade, ICB therapy resistance. In order to confirm that targeting this pathway can overcome the immune checkpoint blockade, ICB therapy resistance, again, we inject the breast cancer cell 41 cells into the biopsy mice and treated the mice with SUV39H1 inhibitor called F5446 alone, or combined with anti-PD-1 antibody, which is a major type of immune checkpoint blockade ICB therapy. As consistent with previous other study, we find the 41 tumor itself were resistant to this anti-PD-1 antibody, as shown here in the blue line when you compare to the black line. In contrast, the SUV39H1 inhibitor F5446 alone can show some anti-tumor effect, as shown here in the red line, and the combination therapy by using SUV39H1 inhibitor plus anti-PD-1 antibody can exhibit superior tumor shrink, as shown here in the purple line. Taken together, this data suggests that targeting the FBX44 SUV39H1 pathway can restrain the tumor growth and also can overcome the immune checkpoint blockade therapy resistance. All right, so this is a summary for this study. So here we identified a F-box protein called FBX44 that can recruit SUV39H1 and to silence the repetitive element in the cancer cells. And those repetitive elements include line, ERVs, and satellite repeats. So I guess the most exciting part of this study is that we proposed a novel biomimicry based strategy for the cancer immunotherapy, in which targeting this signal pathway could reactivate the repetitive element and then to induce the interferon alpha beta signaling and the downstream signalings to achieve enhanced tumor immunogenicity and overcome the anti-PD-1 therapy response. So I guess our study can provide a basis for the development of the SUV39H1 inhibitors for biomimicry-based cancer immunotherapy drugs. And also for due to time limitation, I will not talk about the other part of this study, targeting this pathway also can induce the replication stress and the DNA damage that also contribute to the cell death of the cancer cells and restrain the tumor growth. All right, now let's move on to the second part of my talk, targeting the cancer stem cell signaling to enhance the cancer immunotherapy. All right, so we know that in the tumor microenvironment, there are a bunch of different cell types over there, including the cancer cells, cancer-associated fibroblasts, and some other types of immune cells. And as I mentioned earlier, for the cancer cell part, there are at least two types of cancer cells, regular cancer cells and cancer stem cells. So just like the normal stem cells, cancer stem cells have the features of self-renewal and differentiation, which could dramatically promote the tumor progression and also the therapeutic resistance. The cancer stem cells have been identified in a bunch of different tumor types, including the brain tumor, breast tumor, colon tumor, and also the blood cancers like AML, just to name a few. All right, so this is our current research in the lab studying the glioblastoma or GBM stem cells and their interaction with the tumor immune microenvironment. First, just a little bit of background about the GBM. We know that GBM is a deadly brain cancer, and in clinic, the current therapy failed to prevent the tumor recurrence because those standard therapy, including the chemotherapy and the radiotherapy, they only can kill the differentiated GBM cells. But the GBM stem cells in the tumor, basically they are resistant to all those kind of standard therapy. Based on that, a lot of scientists, they are trying to develop the GBM stem cell based targeted therapy to eradicate those GBM stem cells. However, most of those explorations have been failed to reach the clinic, partially because they only focus on the GBM stem cell signaling itself, but overlook the complex interaction between the GBM stem cells with the GBM microenvironment. Indeed, the GBM microenvironment could prevent the GBM stem cells from the therapeutics as well as immune surveillance by the immune cells. And we are very interested in the interaction between the GBM stem cells with the GBM microenvironment, and we also thought that is a largely unexplored cancer research field. So currently in the lab, we are studying the interaction between the GBM stem cells with other cell types in the GBM microenvironment. And in the for the immune cell part, we especially interested in NK cells due to their innate ability to eliminate the cancer stem cells. And compared to T cell, the NK cell should desired safety and also has a potential for off-the-shelf manufacturing in the future. So we believe that the NK cell immunotherapy has a promise as a next generation effective immunotherapy platform. All right, so this is one of our project ongoing in the lab. By using functional genomics screening in patient-derived GBM stem cell models, we have identified at least 3 genes that can specifically regulate the GBM stem cell self-renewal or GBM stem cell stimulus, and at the same time, those genes can regulate the GBM stem cell sensitivity to the NK cells. And those 3 genes include histone methyltransferase enzyme and 2 largely unexplored E3 ligases. So in order to explore the underlying mechanisms behind these preliminary observations, we are addressing some questions. For example, how those genes express proteins can maintain the GBM stem cell stimulus. Number 2, how their targeting can enhance the GBM stem cell sensitivity to the NK cell immune surveillance. So for those aims, we use different strategies like in vitro and in vivo experimental technologies. So we believe that the study of those 3 targets can provide the further insight into the cancer stem cell biology and also the GBM pathology, finally leading to the development of next generation treatments for the patients. All right, so this is a therapeutic application for our study. As you can see here, under the standard therapy, including the chemotherapy and the radiotherapy, the GBM bug tumor could be destructed. But the GBM stem cell in the tumor, here I labeled in red, basically they can survive and they can self-renew and repopulate the GBM tumor, finally contributing to GBM recurrence. And our strategy is that targeting those 3 novel targets in the GBM stem cells has the potential to stop the GBM stem cell self-renewal and at the same time can attract functional NK cell towards them to achieve 1-2 punch to the GBM stem cells. So we think the combination of those 3 targets, targeted therapy with adoptive NK cell transfer immunotherapy, and also we are also in the middle of developing CAR NK cells, may eliminate the GBM stem cells and finally achieve sustained GBM eradication. Okay, so I would like to take this opportunity to thank our great lab members for their hard work in the lab. And also I would like to thank our collaborators as listed over here. Finally, I also would like to thank the fundings and awards that support our research and my career development over the past several years. Thank you, everybody.